The Phototransduction Pathway. Phototransduction is a process that occurs when light hits the area at the back of the eye known as the retina. The retina is able to transform the light that hits the back of the eye into a signal the brain can understand. This is done through a process within the photoreceptor cells known as rods and cones. In this presentation, we're only looking at rods, but a very similar process occurs in cones. When unstimulated or in a dark setting, these cells are in a depolarized state. This is caused by sodium and calcium influx into the cell through non-selective CNG channels which are activated by cGMP. In this demonstration, we are not showing calcium, however, it is also entering the cell. When depolarized, the cell will release glutamate onto a bipolar cell. There is a protein within the photoreceptor cell known as rhodopsin. Rhodopsin is made up of two molecules, opsin and retinol. When a photon hits retinol, it undergoes a conformational change from the 11-cis configuration to all trans. It unbinds from opsin, which causes opsin to change into metarhodopsin 2. It then binds to and activates the G protein transducin by turning its GDP into a GTP. The alpha unit of transducin will then activate photodiesterase. The binding to PDE allows it to hydrolyze cGMP to GMP. The cGMP attached to the CNG channels eventually fall off, and with a lower level of intracellular cGMP, the CNG channels will close, and sodium will not be able to enter the cell. Potassium, not shown in the diagram, is also leaving the cell, which causes it to become hyperpolarized. Once the cell hyperpolarizes, voltage-gated calcium channels close, causing glutamate to stop being released onto the bipolar cell. This will then cause a different signal to be sent based on if it's an on or off center cell. Recovery stage. Rhodopsin is phosphorylated by rhodopsin kinase. The phosphate group allows arrestin to bind, which prevents it from activating other transducins. Hydrolyzed GTP will convert back to GDP, which causes the alpha unit to dissociate from PDE. cGMP levels also have to be restored. This happens when guanylate cyclase is activated by GCAP. This activation allows it to synthesize cGMP from GTP. This will cause the CNG channels to reopen. The cell will become depolarized again and will continue to release glutamate onto the bipolar cell. Finally, retinol is restored to its original conformation over the next few minutes. This will restore the rhodopsin complex and make it able to react properly to light again.